All right, so let's begin. Thanks to everyone for sticking with us. We're going to have our second and last keynote of the day. So we're extremely excited and honored to have Daphne Koller here with us today. Daphne is one of the giants in the field of AI and has done so many things and received tons of awards, including MacArthur and PKs, and I won't go on and on. Academically, I think she's best known for her work on probabilistic graphical models and computational biology. She used to be a professor at Stanford. Her office was just over there before the remodel. And she inspired a whole generation of researchers. I remember reading her book, actually before it came out in 2005, and learning about graphical models when I first entered the field. She's also well known for co-founding Coursera and pioneering online education back in the day. And for the last 7 years, Daphne has pivoted back to biology and biomedicine in particular. So she's a CEO of insitro, working on using machine learning to discover new medicines. So I'm really excited to learn about what she's been up to lately. So please welcome Daphne to the stage.
Thank you, Percy, and thanks to everyone for being here. It's exciting and kind of weird to be back after so long. So, but I'm excited to tell you about the work that we've been doing and where we hope it's going to go. So. The fundamental premise behind the work that we do at insitro is that human biology is really, really complicated. I've worked in many domains throughout the course of my career, and biology is probably by far the most complex domain that I've had to tackle. And it's not something that the human brain will really ever be able to understand, which is why we create these reductionist models that are like linear pathways on a blackboard, and then we're surprised when the hypotheses that come out of that don't actually work in the clinic. And so the premise behind what we do is that in order to tackle this domain, we really just need to first collect a very large amount of data at unprecedented fidelity and scale at different levels of biological granularity, and then let machines do what they now do much better than people, which is understand the subtle patterns in these data, help us redefine the heterogeneity and complexity of human disease, and identify effective intervention hubs that might give rise to therapeutics that work in the clinic. And so the data that we collect comes in two different forms that we think are very synergistic with each other. On the one side, we've built out a really unique automated lab environment that generates cellular data from human-derived systems with incredible amounts of genetic variation and measured with multiple different high-content data modalities that are unbiased and so are not anchored to the preconceptions of a biologist of what matters and what doesn't. On the other side, we collect data from humans because while we can intervene and do experiments in cells, what we actually want to cure is humans, hence the synergy. And in humans, similarly, we collect very large amounts of high-content, unbiased data, imaging being a primary focus, but also different kind of omic modalities. And we put those data together to try and understand the biology of human health and disease. And so in order to do this understanding, we leverage machine learning. So we use machine learning to basically learn a representation of human biological state at these different levels of biological scale. So this is basically representation learning. I used to have to explain self-supervised learning and embeddings to audiences. I don't have to do that anymore because everybody is now an expert. And so we basically use these approaches to learn a language of biology that is much more crisp in terms of understanding some of the amorphous and fuzzy notions of disease and something that's much more crisp and actionable. And in order to get to action, which is effectively an intervention in a system, we need to get to causality. And one of the very few mechanisms for causality in biology is human genetics. So genetics for us comes in two forms. In our cellular systems, we get to intervene genetically in our cellular systems via these incredible technologies such as CRISPR. In human genetics, of course, we can't do that, but we leverage the natural genetic variation of a human population in order to understand the cause-effect situation, relationship between genotypes and phenotypes. And so that in turn gives us a set of what we hope are translatable targets, which are these high confidence genetic interventions in a particular patient population that is defined similarly via machine learning to define the appropriate group of individuals that are best tackled with this intervention. And then, of course, we have to turn that into actual molecules that intervene in that gene. And so we then do drug discovery to modulate that target in that patient population, which give rise to what we hope will be a pipeline of drugs that will bring benefit to patients. The reason why we believe these two approaches, cellular machine learning as we call it, and clinical machine learning, are synergistic is because, first of all, one is an intervenable system and one is a much more translatable system, but also they allow us to interrogate diseases across a spectrum of genetic architectures, where on the one side we have diseases that are where there is a fairly clear genetic driver or one or more of disease, and we can engineer those into our cellular system and understand the effect of that genetic change at the cellular level. And so that allows us to interrogate those diseases. On the other side, we have diseases that are much more polygenic and amorphous. Those, however, are often much better powered for discovery because they're much more frequent in the population. And so we are able to use human genetics to interrogate those diseases. And we find that there is actually a sweet spot in the middle where there's diseases that are quite polygenic, but at the same time have high penetrance, even if rare genetic drivers. And those allow us to actually bring in both of our tools towards understanding those diseases. And now we are gaining a realization that those are actually the majority of diseases as opposed to the two extremes. Okay, so the therapeutic areas that we're in are metabolic disease, neuroscience, and oncology. And I was thinking I would tell you about all three of them, but there's just not enough time. So I'm going to give you a vignette from oncology and a vignette from neuroscience, which illustrates the two extremes of those technologies that we're deploying. So I'm going to start with oncology, where we really leverage the clinical machine learning capabilities that we have. And in oncology, we basically are leveraging foundation models on a data modality that is very abundant in oncology. Every time someone comes in with a suspected tumor, the first thing that one does is you take a biopsy sample, you put it under the microscope, you stain it with these two reagents. It's called H&E. And it basically exists for almost every solid tumor patient anywhere around the world. It's incredibly high content and multi-scale. It goes all the way from features that you can see within a cell, through cell-to-cell interactions, all the way to much more anatomical features like vascularization. All of those are visible in those images. But when you give it to a pathologist to review, they typically boil down these images of billions of pixels into like 3 numbers. And that's clear that there is a ton more information that is available within them. And so what we've taken on as an effort is to really learn the language of histopathology as a foundation model and then use that to impute additional modalities that are much more interpretable and actionable and give us potential targets. And I'll show you how we do that. So first of all, again, in this audience, I probably don't need to explain this notion of self-supervised learning in order to define a low-dimensional embedding, and the fact that once you have a low-dimensional embedding, you're able to then layer on smaller data sets that have additional covariates that are potentially clinically relevant or biologically relevant, and where you would not be powered to see associations in the original space, but because you have this very concise but semantically relevant representation, you're able to identify these associations and utilize them. So with that self-supervised learning model in hand, let me first show you that, that there is, in fact, a tremendous amount of information that remains, clinically relevant information that remains in these histopathology images that is not currently being deployed. What you see here is something called the Kaplan-Meier curve. Kaplan-Meier curve, the x-axis is years from diagnosis. The y-axis is % of people left alive. And so that curve, unfortunately, goes down and to the right. And what you see here is on the left side is the stratification between high-risk and low-risk patients in a model that was trained using the standard clinical covariates that a clinician would use, things like age and sex and subtype and so on. On the right-hand side is what you get when you add to that set of clinical covariates the information from the embedding. And you can see that you get a much better stratification with a much better goodness of fit and a stronger p-value. So that is nice, but what is perhaps, I think, even more striking is this one, which is a graph that shows on a per-patient basis the risk score based on which the Kaplan-Meier curve was stratified. The x-axis is a clinical risk. The y-axis is the embedding risk. And what is noteworthy is that the correlation is 0. which basically suggests that the embedding is picking up on factors in the pathology sample that are completely invisible to the human eye. So that's interesting, and if we were interested, and we are in fact in prognosis and potentially risk stratification, this is a great start. But what we're interested in primarily is identifying new intervenable targets. And so how do you turn images into targets? And so in order to do that, what we've decided to do is to overlay biological covariates that are molecular entities that are well-defined and potentially intervenable as targets. So what you see here is basically the imputation or prediction of genetic defects in these cancer patients that happen as a result of cancer. Cancer is a disease of genomic instability. And so the DNA changes as part of the cancer process. And those changes are quite clinically significant. And what you see here is that from the same embedding that we trained in a self-supervised way and that we stratified risk in the previous slide, you're able to predict these genetic changes with an accuracy of 90 to 95%. So basically, by looking at a slide, you can say this patient has this genetic lesion versus this other patient, something that no clinician can really do. So that's nice, and in order to basically help train the clinician, in order to help them understand what it is that the machine is picking up on so that they have confidence in the predictions, we've used technologies like generative adversarial networks to create a deep fake, if you will, for histopathology. So rather than generating images of movie stars, we generate images of pathology slides, and what these are is basically traversing in the embedding space from, say CDH1 negative, no genetic variant, to CDH1 positive, yes genetic variant, keeping everything else constant, so just moving along this disease axis. And you can see that the slide changes, but in a way that really homes in on the changes that are caused by this particular genetic variant. And what you see on the right is the post hoc interpretation by a pathologist of what it is that is causing. So this is not something that was given as part of the training, but the pathologist can look at this and say, oh yeah, I can see that there is a histological progression from ductal to lobular carcinoma. And that makes sense to me as a biologist. And so this is a way to help train pathologists and help convince them in the validity of our results. So this is great, but these genetic variants in cancer are quite well recognized, and they rarely form novel targets because people know about them. And so in order to get to novel targets, we've elected to use a different technology that was actually developed here at Stanford by Howard Chang's lab. It's a technology called ATAC-seq. I'm not going to define the assay in this audience, but basically just say that it's measuring the openness or closedness of regions of the DNA. So DNA kind of coils around itself. If it's open, it means that it's very much likely to be active, and if it's coiled together, then it's not. And this assay called ATAC-seq was devised here at Stanford about a decade ago, and it measures this chromatin activity, and it turns out to have a number of important repercussions about understanding activity of regions of the genome, not only for protein coding genes, but actually much more broadly than that. So that's the plus on this assay. The downside of this assay is that it's quite finicky and very sensitive to how samples are handled clinically. And so it's very unlikely that this will ever be used wholesale as part of the standard of care. But the good news is that here at Stanford, about 5 years ago, the same lab, Howard's lab, took 400 samples from cancer from the Cancer Genome Atlas, TCGA, and they ran ATAC-seq on those 400 samples. Now, 400 samples sounds maybe like a lot, but it's not really a lot for machine learning, especially because this was a pan-cancer experiment, which means that the largest sub-cohort within those 400 was 30 breast cancer patients. And 30 is really not a very large number if you want to understand what's going on there. It's not really powered for any kind of, say, a survival analysis. So the question that we asked ourselves is, well, if we can impute genetic variation in the genome, can we impute the activity of individual genes in the genome? So basically, can we impute ATAC-seq, which at this point would basically give us the ability to take those 400 samples and convert these to 11,000 samples worth of ATAC-seq data, admittedly imputed ATAC-seq data, but still. And when we started doing this, we talked to a bunch of Stanford colleagues and said, yeah, good luck with that. No way that's going to happen. And so the answer is you can kind of do that, not completely, but partially. So about 1% of the genome, and this was actually the earlier methods, we've improved them since, are imputable with very high accuracy. And when I say imputable with high accuracy, I mean on held out test patients, of course. And with an R-squared of greater than or equal to 0.5. And you can see what that means both visually on the right-hand side of the slide. You can see that the left part of that shows the actual attack-seq. The right side shows the imputed attack-seq. They're visually quite similar to each other. And the scatter plot at the bottom basically demonstrate that those are, in fact, very highly accurate, imputed with very high accuracy. Which means that we now have 11,000 patients worth of attack-seq for this 1% of the genome, which is actually a very significant 1% because it has histological correlations. Since then, we've considerably expanded the set of histological samples that we have. We have close to 100,000, so we have way more than what we showed here, but this is a good example. And now you can basically start to ask questions like the openness or closeness of which gene, that is the activity of which gene, is most strongly associated with survival, something you would not be powered to do if you had 30 patients. So this is an example for triple-negative breast cancer, of which there are about 10 in the original 400 samples that we started with, but now we have several hundred samples with triple-negative breast cancer. And you can now basically say that there are samples that the openness or closeness, the activity of a particular gene, incurs a hazard ratio change of about 2 for these genes over here. And that is a very significant number. And some of these targets are eminently, they're novel in triple-negative breast cancer. They've been implicated in other cancers. That gives you confidence around the causal role that they play and are potentially really interesting new drug targets. So this is basically kind of a whirlwind view of what we're doing with clinical machine learning. And what you see here is that we have this kind of like virtuous cycle where we onboard more and more high quality data. We continually derive and expand the set of things that we can impute that allows us to learn ever richer and more granular phenotypes and identify genetic drivers that are causal. So now moving on to neuroscience and some of the work that we've done there. So here we're going to go to the other extreme. We're going to use our cellular machine learning, and we're going to use it in the context of a monogenic disease, which is called tuberous sclerosis complex. Not many people have heard of TSC, but it's actually about the same prevalence as, say, cystic fibrosis, which a lot of people have heard of. So about 50,000 US patients, about 1 million worldwide. This disease is a pediatric tumor that presents typically in the brain, and when it does that, it gives rise to refractory epilepsy, by which we mean babies that have 30, 40 seizures a day. When you do that, it's not good for your brain, and it often gives rise to intellectual disability. We picked this disease to begin with because in addition to having a reasonable signature in cellular morphology, which we'll show in a moment, it also has a known reversion, a drug that effectively reverts that disease. It's a rapamycin. It's a form, some of you might have heard of that drug, but it's only effective in 50% of patients, so there is still a significant unmet need. So here we basically create an in vitro cohort of healthy and diseased patients. We start out with a set of wild-type induced pluripotent stem cells, which are these cells that can turn into any lineage in the body. We introduce the disease-causing mutation. In this case, we knock out the gene TSC1 or TSC2 whose loss of function causes the disease. We take a foundation model that was trained on these cortical neurons, which is something that we've also built, and then we overlay on top of that a disease axis that goes from sick, which is the knockout, to healthy, which is the wild type or unmutated cells, and that defines a disease axis for us that we now look for reversions across that disease axis. And so first, before we look for reversions, we validated the disease axis, because when you have these mysterious machine-learned models, you really want to convince yourself that the model is learning something that is meaningful. And so here we had a positive control for reversion in the form of rapamycin. And what you see here is a rapamycin dose response, which basically says the more rapamycin you give, the further the reversion across the disease axis, which gives you conviction that the disease axis is actually going in the right direction. The other form of validation that we did for this is we took human brain samples from TSC patients and controls, and we asked whether the model model that we learned on our disease axis in these, in vitro neurons actually separates the bulk RNA-seq, the bulk transcriptional profiles from human patients versus controls, and you can see almost basically perfect separation. So that gave us conviction that this disease axis is actually a meaningful one that captures meaningful disease biology. This was for transcriptional data. We did the same type of experiment for morphological or imaging data from those neurons. Again, giving ourselves conviction that this disease axis actually means something meaningful to the patient. And so with this disease axis in hand, the question is now, how do you screen for reversions? How do you search the genome for things that revert the phenotype at a reasonable scale? And for that, we built this fairly challenging platform that does what's called a pooled CRISPR screen. This is one of our automated equipment. It's pretty sophisticated and hard to build. You take a pool of cells, you introduce which are all the same, you introduce a bunch of CRISPR guides, each of which is different, all together in a pool, at a number such that each cell on average gets one guide, which means now you have a pool of cells that are genetically distinct. Each of them had a different gene knocked out, but you don't know which cell got which guide. Now you can go ahead and image those cells, phenotype them, and get a lot of really interesting imaging data, but you still need to tie it back to the genetics. And so now you fix the cells, and you basically sequence the barcode that is attached to each guide. So we built a sequencer inside a microscope. And that allows you to do basically thousands of permutations in a single plate, which is which at a single cell level, which is a pretty unique capability. Now, this sounds, great, but in order to really build this, it required a tremendous investment in automation, in chemistry, and a tremendous amount of machine learning, because you can't even segment the cells, especially neurons, which are kind of all over each other, from each other, without the use of machine learning. And so this is a very significant build that, in order to get this to work. With this, we now have a way of screening, basically changing each of those cells and identifying the nature of the reversion, or the nature of the change, and does it revert along this disease axis that we validated earlier. And so we did both a transcriptional screen as well as a morphological screen. So one of them used gene expression as a readout and the disease axis. One of them used cell morphology. Turns out that these are correlated, but still reveal quite distinct biologies, which is actually quite reasonable. So it gave us a set of targets that we then 76, which we got. Okay, now let's see which of them actually make sense. And so the way that we validated those targets is first by testing their validity in human genetics. So this is a fairly well-validated fact at this point that targets that have support in human genetics are much more likely to be successful in the clinic, 2 to 3x times more likely. And so we looked for each of those genes, is there a human trait that this gene is associated with? And fortunately, 55% of them actually had a strong association to a trait that is relevant to TSC. That is, it's the same type of phenotype. And the vast majority of those were actually in neuronal traits, and specifically in epilepsy, which was a really exciting finding because there's not that many epileptic genes. And so the fact that so many of the genes that came out of our screen had an epileptic correlate is a very strong validation of this being relevant. The other thing that we did was that we developed a fairly challenging translatable assay for measuring whether, in fact, the inhibition of each of those genes causes an inhibition of an epileptic phenotype. So in order to do that, you have to take neurons, you have to grow them out to 50 days. That's a fairly complex experiment. And when you do that, they start to fire, and they start to fire in tandem. So even in this, they're all firing at the same time because they talk to each other. So they kind of learn to fire in a synchronous way. And what happens is when you have these genes that have this genetic variant, this disease, and in fact for other epileptic diseases as well, that genetic change causes them to fire in a very dysregulated way, and you can see that in the diagram in the middle, and in a way that is quite concordant with what happens in human EEG for those patients. So it's a very close mirror in the dish to what you see in human disease. And so we took initially 14 of the hits that came off of our screen, and we put them into this 50-day assay. And remember, the screen was on 10-day neurons, and they hadn't even started to fire in tandem. And we were frankly shocked that about 50% of those 14 genes ended up giving a reversion of the epileptic in vitro phenotype, that misfiring phenotype, in a way that was actually concordant with the amount of reversion that you get from rapamycin. And so for those of you who are interested, the red is, for example, the firing rate that you get for the TSC knockout genes. You can see that it's considerably higher than the wild type, which is the green that's next to it. The blue is what you get from rapamycin, so reversion that's very close to wild type. And what you see in the purple is what is called a dose response. The more of the drug that corresponds to our gene you give it, the closer it gets to wild type, which is a very significant source of conviction building that this is truly a disease-reverting gene. And so we are now we've selected a subset of those genes that we think make for good drug targets, and those are currently in development for us as drug targets. So this is, we started out with TSC. We're now working on a much more complex disease, ALS. Many of you are familiar with ALS, also known as Lou Gehrig's disease. ALS is one of those diseases actually is at that intersection, in the sense that there's a bunch of high penetrant genetic variants, familial variants that are quite significantly causative of ALS, but at the same time, 90% of the cases are sporadic, where the genetics are much more diffuse. And basically, we are deploying this technology to try and interrogate the heterogeneity of ALS and to understand what reversion looks like. And so in order to do that, we have built the largest collection of isogenic iPSC lines that capture both the sporadic and the familial genetics of ALS. We have an automated process for taking these stem cells and converting them into motor neurons, all of the phenotyping and screening capabilities that I showed you, and we have actually just got to our first set of targets from these ALS lines, and we're very excited about that. So summarizing this piece, the other part of our technology stack is these human-derived biologically relevant cellular systems at scale with very high content multimodal phenotypes that help unravel the heterogeneous effect of genetics, and machine learning to identify cell states that align with human clinical outcomes, and then using genetic screens to identify modifiers that revert a sick state back to a healthy state. So I'm just going to end with this like philosophical note. I hope I can take one more minute, Percy, which is, as I embarked on this journey, I thought back on the history of science, and when you reflect back, you see that there's periods in history where a particular discipline makes a very significant amount of progress in a very short amount of time. And you can think about late 1800s chemistry with the uncovering of the periodic table, early 1900s, of course, physics with the connection between energy and matter, space and time, and then, of course, computing in the 1950s. And then in the 1990s, there was an interesting bifurcation. There is data / machine learning / AI, which is really something that began back then. And then on the other side, quantitative biology, which is the ability to measure biology at unprecedented fidelity and scale. And those two disciplines have been evolving in parallel for quite a while. And I think the time that we're in now is an era where those two are going to come together and create a new field. And I call that field digital biology because it is the ability to read the biology digitally at this incredible fidelity at unprecedented scale, interpret what we see using tools of machine learning and AI, and then write biology using techniques like CRISPR and combinatorial chemistry and all sorts of other things to make biology do things that it wouldn't otherwise do. And I think that has tremendous repercussions in human health, but also in the environment, in energy, in biomaterials, and sustainable agriculture, and many other disciplines that will help make our world a better place, which is why I think it's a really exciting place to be. So thank you very much.
Great. Thank you, Daphne, for that wonderful, inspiring talk. Really impressive work. If you have a question, raise your hand and the mic will come around. Maybe I'll start with a question. So you talked about the whole spectrum from running the experiment and collecting data, building the model, training the model, and interpreting the results.
Yeah.
Can you say a little bit more about where the bottlenecks are. Are we just swimming in data and we just need to build better models? Are we still data bound in some regimes?
I think the answer is yes on both.
Okay.
So, I mean, a single experiment like this generates hundreds of millions of cells. And so the amount of data that we generate is huge. If you think about even a single histopath image, it's billions of pixels, and we have many, many of those. And so in some sense, we're swimming in data. But on the other side, there's the space of different biologies that one can interrogate is effectively infinite. You can introduce different genetic variants, you can introduce combinations, you can add additional chemical exposures, you can grow the cells out for longer, you can put cells together with other cells. It just boggles the mind how many different ways there are to take these models. And so, yes, we have a lot of data on the one hand, but yes, it will never be enough on the other.
Yeah, I thought that might be the answer.
Yeah, sorry.
Questions? Otherwise, I'll. Okay, in the front here, where's the mic?
So thank you for a wonderful talk. So maybe a question in two parts. So the validation of the targets, as you talked about, will appear to be all in vitro. Did you look at model organisms to validate them in vivo, number one? And part two, so there has been this argument in drug discovery for a long time. Do we need new targets or do we need new modulators of the targets? So the discovery of these targets, in your opinion, does that allow us to look into chemistries which will be easier to modulate them?
Yeah, so both great questions. So on the first one, yes, we are currently looking at model systems. The good news is that for the TSC disease, because it's a monogenic disease, you can actually create that lesion in a mouse, and it actually exhibits symptomology that is concordant with that of a human. So those mouse experiments are currently ongoing. Those are very fragile mice, so the experiments are hard to do. I didn't show you the work in metabolic disease. There we're dealing with a different type of indication having to do with fatty liver that arises from obesity. Those mice are very robust, and so those experiments are easier, and they're looking quite good for some of the targets that emerge from there. So I would say, that having been said, I would caution against just immediately devolving to animal models, because for some diseases they're reasonable. For other diseases, they are quite distant from the human biology, and you have to treat them with great caution. In terms of the, you're exactly right about, you can think about better, there's a set of targets that we have conviction around already, and we could intervene at them with new chemistries, and there's certainly people working on various forms of chemistry for these for the old targets that we have. At the same time, honestly, most of the drugs that go into the clinic and fail in phase 2, which unfortunately is the majority, fail because of lack of efficacy. It's not because and it's not because the drug doesn't work in modulating the target. It's because the target doesn't do what we thought it did. And so I think there is a huge value to uncovering targets that are novel, potentially with larger effect size, potentially that admit easier chemistries.
Okay. Let's see, we'll go maybe back there for a question.
Yes, thank you for the talk. I was wondering about the data collection process. So obviously it's very complicated and presumably quite expensive. After you sort of mine it for actionable insights, if you decide that something isn't like essentially monetizable, maybe it's not commercially feasible or it's just unhealthy, risky for a patient, and you reject that as like not feasible, would you consider open sourcing that data to the community? Because it might have scientific value about trying to understand biology at a fundamental level, even if it's not actionable or commercially viable.
So we have recently posted on bioRxiv a paper around the POSH platform, which is this pooled optical screening platform, and it came with a data set that focus on cancer lines, A549 lines. Those are not lines that we're currently actively working on because we're not using cancer lines for oncology work. We're using primary human data, and so we released a fairly substantial data set with that paper, and so the answer is yes. Drug discovery is a long journey, and so some of the other data sets that are still in progress are not yet something that we can release, but we will certainly consider doing that as those data sets become less encumbered. And the A549 data set is really cool. Tons of biology.
Yeah, I was actually going to ask this something very similar, which is if you look at the landscape of different biological data sets, what does the ecosystem look like? How many data sets are in the public domain versus
I mean, there is a number, and they come in different forms. So in terms of cellular data, there is actually a fairly significant amount of RNA-seq data because the Human Cell Atlas supported a lot of that. So I think that's an area where there's a fair bit. They don't include perturbations. They are just cells in their natural state. There is a much smaller amount of perturbed cells. On the morphology imaging side, the Broad has released a data set. We've just released a data set. Recursion has released a data set, but that's pretty much, I think, there's not a lot more beyond that. On the human data, I think things are more complex, and also there's, a small number of data sets that I would consider to be high quality, and then a whole bunch of ones that I would be wary of.
Okay. So this is a related question I'll take from the online audience. What should the main difference in the roles between industry and academia? Obviously, you've been in both locations. You are in industry right now, and obviously there's a bunch of related work happening in academia. Is a natural complementarity there?
I think there's certainly some really exciting work happening in academia on pushing the boundaries on methods development. We do that as well, but we have a much higher burden on actually making those things not a use once, post on GitHub, move on to the next paper, but something that is actually a robust, reusable infrastructure. So it takes longer for us to deploy and robustly a method. And then, of course, I think some academic institutions have dabbled in actually making medicines. By and large, it's been a challenging journey for an academic institution to do that. And so I think as you start to think more about the translatable elements of this, that generally works better in industry.
Okay. We have time for maybe two more questions. Rishi?
So maybe I can also ask a question given the many different places you've been. So one thing you mentioned earlier in the talk is about using your work for training pathologists, I think, if I remember the example correctly.
Educating, I think, is probably
right.
better.
Right. And I found the example very interesting of using the scan to generate the example and so on. So I wonder if there's any crossover between what you saw as an educator versus what you saw in this context of educating pathologists, or if there's any relationship between the two.
I mean, I think the current educational process for clinicians in general this is what we look at, and it's fairly, very well laid out because that gives you some level of consistency among clinicians, so there's a real rationale for that. But I think what these methods allow you to do is to look not under the lamppost, and sometimes that actually uncovers novel biology. And I go back to a paper that I wrote, one of my last Stanford papers, which preceded deep learning, but did an unbiased machine learning model on the tumor microenvironment. And it basically discovered the importance of the stromal tumor interaction that had not been previously appreciated. And because it didn't rely on just trying to predict what pathologists had been taught to predict, but was taking a much more unbiased perspective. And I think that there is a tremendous value to sort of sometimes just letting things loose and see what comes out.
Okay. Maybe one final question in the front. Okay.
Thank you so much. So you give very great use cases of therapeutic, right? I'm kind of curious about diagnostic in rare disease, undiagnosed, or any of subtyping of complex disease such as diabetes, how you can leverage this data.
Yeah, and that's a great question. We are not in a diagnostic space, largely because as a startup, you kind of need to focus, and whereas a lot of the underlying technology stack would be similar, the go-to-market strategy is completely different for therapeutics and diagnostics. So we've decided not to go there. I think that when you look, for example, at some of the work that we're doing with histopath, it clearly elucidates heterogeneity of the disease and can help create more high conviction diagnostic predictions, can also identify subtypes in ways that are often very hard for a clinician to do. I think there's a tremendous opportunity there, and I hope someone ends up, there's a number of companies in this space, and hopefully they will be able to make a difference there.
Okay, I think that's all the time we have. Let's give Daphne a round of applause.